We have one more talk before break. This will be presented by Karen Mock. Karen is an assistant professor in the Department of Wildland Resources here at Utah State. She obtained her PhD from Northern Arizona University and her master's from Colorado State, BS from University of Texas. And she's a well-known geneticist, and she's going to become a better-known Aspen geneticist in the near future. Title of her talk is Using Genetic Tools in Western Aspen Ecology, Conservation, and Management. Karen?
Okay, I'm happy to see that everybody's running out the door, even though this is a genetics talk. So I'm Karen Mock. My co-author is the first person I'd like to acknowledge here, Carol Rowe. She's my lab manager, and I will admit right up front that she really does all the work. Carol and I run a conservation genetics lab at USU, which works on a very wide variety of species, some of which are shown here, and one of which is aspen. In fact, aspen is the only plant species that we're working with in our lab, and it's presented us with some of the more interesting results. So what I'd like to do today is first give you just sort of kind of an overview. This will be really boring to this crowd. Just a quick overview of Aspen as a research system, and maybe from a genetics standpoint, and a little bit about our current research trajectory, at least in my lab here at USU, and finally to show you some of our early results, which I think you'll find very interesting, and maybe some of you can help me interpret them. So I also wanted to start with a little terminology. Again, this is old hat for most of you in the room, but I'd like to be able to use the terms ramette and genet in this talk. A ramette being a single stem, a single tree, a single shoot, if you will, and genet being a single genotype. So a genet then may be represented by one or a bazillion ramettes. So a little bit about why Aspen is interesting from a management perspective. First, obviously it's been declining rapidly in many western regions over the past century. It's also a very important browse species, maybe to the consternation of people trying to grow aspen. And aspen stands obviously contribute a disproportionate amount of ecological diversity compared to other tree species, particularly in western landscapes. And genetic diversity within aspen has generally been ignored in management-oriented research, but one prescription, if you will, is unlikely to fit all aspen genotypes. And finally, aspen is a valued component of the recreational landscape in the West. In other words, it's linked to tourism and recreation dollars. Aspen is also an excellent system for evolutionary and ecological work for a variety of reasons. First, it has just an enormous geographic range, just enormous. If you look at this, is the single species that's going all the way from Nova Scotia to Alaska and all the way down to Mexico. That covers just an amazing amount of ecological diversity. But one species is able to find places to live in that whole range, which is really amazing to me. So Aspen is a really good system then for looking at adaptive genetic variation across this huge range of environmental conditions. Another reason that Aspen makes a really good research system for evolutionary ecological questions is that Aspen in particular, and populace in general, is extremely diverse, both genotypically and phenotypically. And this diversity is not just because of its enormous geographic range. Even within a small area, you can see huge diversity in traits like branching pattern, leaf shape, growth rate, suckering rate, phytochemistry, things like that. And you've all seen that. Anybody that's been out in the forest here has seen incredible diversity. For example, Rick Lindroth, who gave a talk here that some of you may have seen about a year ago. He's from the University of Wisconsin. He's finding enormous genetic variants in chemical defenses in aspen that were obtained from a very, very small area. These two stems right here are two different genotypes. They were grown in a highly replicated common garden along with many other genotypes, which was then opened up to deer browsing. And clearly the deer didn't think all aspen were the same. So deer, as molecular geneticists, deer clearly have a preference for certain genotypes, and other genotypes were not preferred. The one that still has leaves was among several of the genotypes that had genetically high levels of defensive chemicals in them, even when they were grown in a common environment. Okay? And as more and more common garden experiments are being done, we're finding this very same kind of pattern, just enormous variance, much more than you would expect in a well-behaved plant species. So this raises some questions right away. How much of this phenotypic variance, when you look around and you see trait variance in these trees in the landscape, how much of that is genetic versus environmental? For example, it could be soil variation, historical variation, something like that. So how much is genetic and how much is environmental? And how is this diversity distributed on the landscape? In other words, are there particular areas in the West or maybe in Utah that have much higher or lower genetic diversity for traits than others? And finally, what difference does this diversity make ecologically? As an example, this is a photo from an area that I think you're going to go by during the field trip tomorrow. Ron's nodding. And this is a clonal boundary that is right by the Swan Flats Road at the top of Logan Canyon. So remember this clone when you're on your field trip, or this stand. In this group of trees here, there are at least 2 genotypes. There are probably more, but I know of at least 2 that are there, or genets that are represented. And in 2005, I haven't been to this stand this year, but in 2005 it was very clear that if you walk into this, you could see the left and the right are at least 2 of the genotypes there. It's very clear that one of these was much more susceptible to a particular leaf fungus, and I couldn't name that fungus, Fred Baker could, but apparently genetic predisposition for susceptibility to that fungus was really marked in one of these clones. I believe it was the one on the right that was really susceptible. So it'd be interesting to see if we still see big differences there. So if you think about this on a landscape scale, then genetic diversity in aspen could actually be linked to things like insect diversity on the landscape, or mammal diversity, or bird diversity. All right, and those are things that we don't often think about. We tend to think of aspen as just being aspen. Genetic diversity could also be linked to understory diversity. It could even be linked to soil nutrient processes, okay? Nutrient cycling, even water dynamics at the watershed scale. Perhaps it's linked to ecological efficiency or stability over time. Ecological theory would predict that. And we don't know if that's true, but it would be an interesting question. So another reason that aspen's interesting as an evolutionary and ecological system, of course, is because it's clonal and because it can reproduce asexually through these rhizomatous roots. In the West, particularly in the Four Corner states, obviously single genets can have many, many ramets, and the clones can be absolutely huge. For example, the Pando clone in the Fish Lake that Bob Campbell was talking about is estimated to be over 43 hectares. This suggests that some of these genets are quite long-lived, and they maintain themselves over these long periods of time clonally. Some botanists suggest that aspen has not established itself sexually to any real extent in the West since the last ice age, 10,000 years ago. Some have even suggested that clones may be as old as 1 million years. Now, those kind of statements get the attention of geneticists. We consider it a challenge. So if you think about clonality, we really don't know a lot about how these particular genets move, how they fragment, and how they may be regenerated over time, especially in the West. For example, this is just a silly cartoon of one possible scenario, but if you start up, I guess I do have a mouse here. If you start up here with a number of genets that were sexually generated, and then if you were to turn off sexual reproduction and let them expand asexually, you would likely find that different genotypes expand slower or faster, and that eventually, if you continued, that some would be eliminated, some would be huge, maybe several would be huge, many would be gone. And if you then follow that, if this is a big landscape and now you come in with some sort of a disturbance, you can imagine a scenario where you might have genets that are split up. So you may have one genet in one place on the landscape and the same genet exists in a totally different place. That would be disjunct. We really haven't done the work to understand how these clonal dynamics work in Aspen, but hopefully I'll be able to convince you that that's an important question. So there are several questions that we're very curious about in our lab. So at USU, these are three of the primary areas that I'm interested in my lab. There are many ancillary questions that I'll try to stay away from. But these are the three main areas. So at USU, there's been a long tradition of working on Aspen, but until now, we really haven't had a genetics component, and that's something that I hope my lab will be bringing to the table. We're already involved in several specific projects, and I'll go through those today. They fit sort of into these three categories: range-wide phylogeography, mutation accumulation in Western clones, and reproductive and clonal dynamics in Western landscapes. So we'll start out with phylogeography. First, one of the things that has not been done yet for Aspen, to my amazement actually, is to describe the evolutionary history of the species. We've done phylogeography on almost everything else, but we've never really had a comprehensive phylogeography done for Aspen. Phylogeography just means the evolutionary history in a geographical context. So this is really important. It really is important. It's an important framework if we start looking at genes, specific genes of adaptive significance. And that's an inevitable direction in research because the populus Trichocarpa genome was recently sequenced, which means essentially that the Aspen genome has been sequenced, essentially. That means that we're going to start looking at specific genes that are of adaptive importance, adaptive significance. We need to have a good, solid phylogeographic framework if we're going to understand the distribution of those genes of adaptive significance on a big landscape. If we have that framework, it allows us to detect the signal of very strong regional or local selection on particular genes. It also helps us understand the geographic movements of Aspen over very long time periods. This map shows our locations, our sampling locations so far, so actually pretty far along with this. We've been collecting Aspen samples for the last couple of years. Many of these collected by people that were just interested in helping out the project. Each red dot here is a location where we've collected between 20 and 30 different genets, hopefully, from about a 10 to 20 mile radius. And we have well over 1,000 samples so far on this collection in a total of about 33 locations. In the next couple of weeks, we should be collecting samples from Mexico. So this is going to be a very respectable sample set. Now, I have a little pitch here. If you look at that map and you say, well, my area doesn't have a red dot in it, I'd really like to hear from you. And if anybody's interested in helping me put more red dots on this map, I'm all about that. So hopefully my contact information is there. So please don't hesitate to contact me if you want your own personal red dot. Okay. So the next question that I'm really interested in, this is one I'm really curious about, is the accumulation of harmful genetic mutations in Western Aspen. And it's not, to my amazement, again, it's not something you hear people talk about a lot. But if you really think that you've got a clone or a genet out there that's 1 million years old, this is an important thing that we should be considering. So one of the big benefits to sexual reproduction in any organism is the ability to purge harmful mutations from a population by way of genetic recombination and subsequent natural selection. All right. But in asexual species, harmful mutations can accumulate; they can build up, and you've got no way to purge them from the population, and so these things start blinking out. So you might expect that could happen in Aspen if you really have a very low rate of sexual reproduction. So because that's supposed to be the case in western Aspen, that we have very low rates of sexual reproduction and the potential longevity of these genets really may be at significant risk for the accumulation of deleterious mutations and the associated loss of fitness. So a third category of questions that we have that we can do something about in the genetics lab is the reproductive and clonal dynamics in western landscapes, which I think would be very interesting. And this is fairly easy to get at with genetic tools. This is the one I'll spend the rest of the time on since we have some preliminary data. And I've tried to line out the basic research questions here, although there are so many others. First, how well do fall and spring aerial photos or other measures of morphological divergence, like personal opinions or things like that, how well do those capture existing genetic diversity in aspen stands? Second, are genetics cohesive? Are genets cohesive at the landscape scale, or are single genets represented in multiple locations? And that goes back to that cartoon that I showed you. How are these things distributed? We really don't know. And finally, to what extent does sexual reproduction occur and contribute to diversity in Western landscapes? And is this sexual reproduction associated with particular landscapes, with particular features, or particular landscape histories, perhaps? And so those are all questions we can begin to get at now. So we've got too many questions and not enough people in the lab, as it turns out. All right, so for focusing then on this third set of questions, we tackled this by finding a study area. We thought, okay, we're going to look at this. We need to define a study area. So we tried to find a study area that was reasonably easy to get to and happened to have a road running right through it. and seemed to have some amount of interesting diversity of landscape. So we chose an area that is at the very top of Logan Canyon, and you can see that. Where's my pointer? If this map doesn't have roads shown on it, but if you were coming up Logan Canyon, you'd go over the top and over to the Bear Lake side. And hopefully you're familiar with that area. Right before you get to the top, you get to the Swan Flats turnoff, which is just a dirt road. That dirt road does a big switchback. Where is that? And then it goes straight north to the Utah-Idaho border. And this area is about 6 sections, this whole area, and we decided that would be a handy area to start looking at some of these clonal dynamics. In retrospect, maybe we should have picked something with a little better described or more interesting landscape history, but I guess you can always do that. This is a much closer picture of that entire 6-section study area. We being a kind of a tattered crew of geneticists and anybody else we could round up, sampled all the aspen in that whole 6-section area on a 50-meter grid. And that was quite an effort. It was right before deer season. Yikes. And this is, every dot here is a sampling point. And the reason you've got different clusters of dots is not because those are different genotypes, but because we call these different polygons, right? So we had one polygon here, another one here, another little one over there. You could think of these as stands, perhaps. Okay. Again, these are just all our sampling points. Here's that you can see down at the very bottom here. This is the little Swan Flats turnoff. These are the samples from that clone that I had showed you earlier. All right. Okay, so that amounted to about 850 pretty impressive for a crew of geneticists, don't you think? 850 leaf samples collected mostly in the fall of 2004. We then took them back into the lab where we were much more comfortable and genotyped them at 7 polymorphic microsatellite loci so far. And we focused first on 5 of the polygons. We thought, well, we can't do everything, so we'll just start somewhere. And I've got that data for you today. These 5 polygons make up 526, so the majority of the samples of those 850. Okay, so these are the polygons that I'm going to be focusing on, and we'll look at each of these a lot more closely in turn. But first, this is our whole study area with those 5 polygons, and I should thank Ron Ryle right up front because these are maps that he generated with my genotypic data, and he saved me a lot of time. Now, we'll be looking at each of these in turn, but right off the bat, we've got an issue here. In just 5 of these polygons with 526 individuals, we identified 103 different genets. This is way more genets than most foresters would have estimated based on morphology and conventional wisdom. And I guess that's the way to say that. It was way more than anybody expected. And this is actually quite a conservative estimate for a couple of reasons. One, any of the genotypes that differed just by a single mutation, a single allele, were pooled. Right? So we took that conservative step, and we still ended up with 103 of these things. 37 of these were single observation genets, which means that we only saw them once, and it suggests that if we had sampled on a finer scale, we would have more genotypes yet. These genets did seem to be rather cohesive and not scattered among the polygons. I know this is a very busy slide, but with 103 different symbols, it's kind of hard to make a real beautiful slide. So bear with me on this. You can sort of see just by looking at the symbols there, each symbol represents a particular genet, and that they did seem to kind of stick to their polygon. We didn't find the same genets scattered all over the landscape. So they were rather cohesive. There were 3 instances out of all of those genotypes where a genet was present in 2 polygons, and 2 of these were right here between these 2. That's probably because we made 2 polygons out of this, and maybe it should have just been 1. So it's not too surprising that you would find 2 shared genets between these. The other one was a little harder to explain, and that was down here. You see these turquoise squares with the black dots, and here they are again up here. Statistically, with 7 polymorphic loci, we do have the power to distinguish individuals. I'm really not wondering to myself whether this is a different genet up here. I'm really convinced that it's the same one. So that's kind of interesting. But there was only 1 of those. That was the exception. Okay, now let's look closely at polygon number 5 here. That's just kind of a locational picture. Okay, these slides are going to get harder and harder, so squint. Here's a close-up of polygon 5 with the genotypes overlain on an aerial photo. And if you look at these, you can see some of these genotypes are quite spread out. Look at the green squares here. It occupies a bunch down here, and here's a few more up here. But we didn't find that genotype anywhere else on our study site, just here. Here's the red crosses. They were pretty spread out. Some of these were a little bit more localized. You can probably pick out a few of those that were a little more localized. We did have 5 single observation genets. And the weird thing about this polygon, can I use the word weird in science? That was really weird. We found 11 genotypes that looked like triploids. Okay, 11 out of 19 unique ones. Okay, that's a bloody lot. And what I mean by possible triploids here is that at a particular locus we found 3 variants. And it was 3 variants. It was clear as day. And some of these had 3 variants at 4 out of the 7 loci. So they really look like triploids. I'll come back to that in a minute. A triploid, by the way, means that you've got 3 copies of the genome instead of 2, which is the more well-behaved form. All right. Now let's look at polygon 8. This is the situation there. 25 unique genets, 11 single observation genets, and 8 possible triploids among these. Interestingly, the triploids did seem to, they tended to be the larger genets. They tended to be represented by more ramets than others. I'm not sure if that would hold up statistically, but we'll find that out. It certainly kind of looked that way. Okay, and again, you see the same pattern. Some of these are spread all over the place. Some of them are really rather localized. Okay. Now, for this polygon, we also had fall aerial photos, and that was nice. We actually have fall aerial photos for the entire area. It's just that they take a while to orthorectify, and we haven't done that for all of them yet. So what happens, now I know this is really bad, but when you put all these bazillions of genotypes on top of the fall aerial photos, now you can start asking the question, oh, can you look at a blob of bright yellow aspen and say there's a genet? Or can you not? And the answer is not, it turns out. If you look at the genotypes here, they really don't correspond worth a darn to the aerial photos, the colors that you see in the aerial photos. Now, I will admit up front that this is probably because of the sampling bias we had. It's still very interesting. But when the crew of geneticists and other folks were out on the landscape collecting samples, we just collected the leaves we could reach, which means we had a very strong bias for short trees, right? An airplane, by contrast, has a very strong bias for tall trees, and so that may be, at least in part, responsible for this disconnect between what the airplane sees and what we saw in the lab. Still, though, it's really surprising that was really a strong disconnect. Now, if you look at this, this is the part of the polygon that's in the lower part of that diagram. If you look closely at that, you might think, well, surely there, that really looks homogeneous, except for maybe this area over here. And again, there are all sorts of genotypes hovering around in here. Maybe they're all just in the understory, but this is really interesting. Okay, on to 15 and 16, which I treated as a single group here. Pretty much the same sort of pattern, 35 unique genets, 16 single observation, and only 1 possible triploid, which is kind of interesting. Okay, same kind of story with the distribution of color. We didn't find a very close relationship there. Polygon 19, just whip through that one. We don't have a real pretty picture of this one, but sort of the same pattern. We had local genets, but they weren't shared with other polygons. Here's our summary data. So overall, there were 3 big surprises. First, we found way more genotypes than we expected. Second, there were a lot of singletons, a lot of single observations, so we may have actually underrepresented the genotypes that were there. And third, we found this high level of triploidy. The triploidy is very interesting because triploids you would expect to have at least a somewhat reduced fertility. Now, in a landscape that favors asexual reproduction, maybe that kind of makes sense because triploids also have greater heterozygosity. In other words, they've got 3 versions, or potentially have 3 versions of each gene, right? So if you've got deleterious mutations coming up, perhaps the triploid state is a buffer against that. I don't know. Okay, so back to our research questions. How well did the fall, and it turns out the spring, also aerial photos capture existing genetic diversity in these aspen stands? The answer is not very well. Second, are genets cohesive at the landscape scale or represented in multiple locations? They're somewhat cohesive. They don't often occur in these multiple polygons, but we really need to start sampling cohorts, the understory and overstory separately, to really get a handle on these dynamics. And finally, is there molecular evidence of sexual reproduction in these western landscapes? And the answer is probably yes. This is a really interesting finding. The allele frequencies are quite similar among these polygons, even though the genets are unique. That is a pattern you might expect if you have sexual reproduction. There are many rearrangements of the same alleles among genets, so we can't just blame this on accumulation and mutations. And we have very high genotypic diversity overall. Still to do, we need to estimate the relatedness. We need to do spatial autocorrelation on these genotypes, because it would be very interesting to know whether the genotypes within polygons are more closely related than between. Okay, that might help us form some conclusions about these landscape processes. I have a lot of acknowledgments. I've been wandering the halls of USU, wringing my hands about Aspen for a long time, and have garnered a lot of support. And I'm not going to read through all these names. You'll be happy to know. But there are lots of USU faculty folks. There are lots of USU students, research people at other universities, lots of agency folks. And I definitely want to express my gratitude for the funding that we've gotten so far on this kind of work, which comes from the Community University Research Initiative, the USU Advanced Program, a little bit from USU Extension, and a little bit from the Forest Service. So with that, I'd be happy to try to take any questions. Yeah.
I'm sort of interested in the sampling method and how that relates to those single genet observations. When you say single genet, you mean one stem that you sampled revealed that genet, but that doesn't necessarily mean that on the landscape there's only one stem.
Absolutely.
That your sampling method. Can you talk a little bit what is sampled at each point, which is 50 meters from the next point?
Yeah, the 50 meters seemed like a pretty close grid when you're talking about 6 sections of land, but when you're really talking about small-scale dynamics, it's really a pretty big jump. So it certainly doesn't mean that there was that genet only existed in one stem and we got it. It means that we only sampled it once.
So it may have been quite large. It's just that
yeah.
We don't know that.
Yep, it's just a matter of probability. So when we went to when we sampled this site, we had pre-assigned sampling points, and we would just walk to them with a GPS unit and sample the closest tree.
So just one tree at one point.
Mm-hmm. Yep. Yeah.
Short tree.
Short trees, yeah. Some didn't seem that short. There were occasions when we couldn't find any leaves that were reachable at all, and in those cases we did actually scrape a little bit of cambium, but there was definitely a bias. It's much easier to extract DNA from leaves than from cambium. So that was our bias. We've actually replicated this sampling design in other locations now, and in those locations we sample big trees and little trees intentionally, and so that should be kind of telling. Any others? Yeah.
Is there a possibility that if you go to these stands that seem to be declining, and they could decline, that you wouldn't find that diversity? Is that
Yeah, that's who knows? Anything is possible, I think, with this species. Certainly, it makes you wonder, doesn't it? That on some of these, especially in these southern landscapes, that you might find that the genets are actually much bigger. Maybe there's a whole lot less sexual reproduction. Maybe there are big genets, and in that case, I would suspect that mutation accumulation could be a very serious thing to consider. That might be a big impact on fitness. That's something I think really important for managers to know about, because no amount of grazing protection is going to protect you if these genetics have a lifespan. Yeah.
Certain genetics have better resistance to disease. I mean, you've been in.
Oh yeah.
I assume you've been inside these stands.
Yeah. Yeah, the differences look pretty pronounced. We don't have as fine a scale sampling as we'd like, but clearly there are some obvious clonal boundaries. You can walk to them. You can say, "Oh yeah, here it is." And then when you begin to look closely, you can. It looks to me, I haven't done the science behind it, but it looks like there are big differences in insect populations, susceptibility to fungal diseases.
Some of the traits that work anyway.
Oh yeah, that's huge. Yeah.
I've got one more.
Yes.
I just had a question as to whether you find that the area covered by a particular genotype, like you have some idea of the length of time that genotype has been on the landscape.
It could, and that would be, of course, there'd be all sorts of qualifiers, but that's, of course, the first thing you think about when you see a genet that's way over here and way over here. You think, boy, it must have been continuous at some point. So, yeah, it does make me think that. Now, that, of course, hidden behind your question is the ability to age clones, eh? And how do you do about that? I think there's one situation where you might actually be able to develop the aging tools for clones, and there are people in several labs that are working on this right now. But what they all lack is a clock. They all lack calibration. You need to start with a genet of known age. Now, one idea that's occurred to me in the past is to look at some of these very young volcanic fields. And some of those do have Aspen in them, and some of them maybe, if we were lucky, we could find one that was 1,000 years old or less. And if we have that situation, you might actually be able to calibrate it, and then you'd be off and running. Yeah. Okay.
Thanks.
Yeah.